going to turn today, the next section, over to the pharmaceutical company pipeline. And this is where Dr. Fargo is going to come back and talk about what the pharmaceutical companies are doing to develop treatments for CMT. So who you should be writing and letting them know that keep up their good work. We at the Research Foundation know the CMT drug development ecosystem. And since Dr. Fargo joined the Research Foundation, he's been immersed in the field. He's been scouting new projects from those currently working in CMT, like Rob Burgess, and also seeking out new people like DTX with their new technology. He has been scouting the ecosystem and funding and bringing grants to the table to be funded. They are funded through the process by looking at, by being evaluated by our scientific advisory board, who looks at it from that basic science perspective, the clinical perspective, as well as the commercial perspective. They look at the whole spectrum to make sure that we're ready for success. In this session that Keith is going to lead, speakers are going to give an overview of the efforts currently underway. So these are all the things that we just heard about from Rob, a deeper dive into that. So traditional medicines, along with genetic and cellular medicines. So welcome back, Dr. Fargo.
Oh, thank you, Susan. I appreciate it. All right, let's jump in. So we're going to have a whole session here on industry research. I'm going to start by talking about something called small molecules, and you notice I put a little plus there, and you'll see why later. And then we're going to hear from Dr. Amanda Hdac6, who will talk about gene therapies. And then we'll hear from Dr. Sung-Chul Jung with a very innovative approach using cellular therapies. So I'm going to go ahead and jump right in here. Small molecules. Why do we even care about small molecules? And you heard some about this, from Will earlier, but I'm just going to reiterate it. We know that these are genetic diseases, so why not simply fix the genetic defect and cure the disease? Well, certainly that is what many researchers are trying to do and working very diligently to do, as you've seen earlier this morning, and there's tremendous promise there. But why do we, if we're doing that, why do we also then need to go after some more traditional kinds of medications? Well, and there are lots of reasons for that. One is that there are many genes and many mutations. And so it may take time to get to genetic therapies for all of them. And in the meantime, if we can treat people with other kinds of therapies to alleviate, halt, or potentially even reverse that disease process, why would we not want to do that? Also, we have to think about treating people after disease onset. So when we imagine the future with genetic therapies for CMT, one of the things that we think about is the timing of that therapy. And will that therapy be as effective in people who already have symptoms? We don't know the answer to that yet for people. We've seen in some animal studies that some of those gene therapies do have some benefit. Some have quite a bit of benefit for people who, or rather for animals who have already developed symptoms, but we just don't know yet for people. And so we have to think about how we can treat people who have already developed symptoms before genetic therapies have become a reality for their subtype. Another thing that I want you to think about is that many genetic diseases can actually be treated by non-genetic therapies. And I'll just give you an example of that. I'm a little bit of a storyteller by nature. This isn't a long story, it's just an anecdote about myself. I have a familial hyperlipidemia, which means that my cholesterol and my other lipids are sky high. As a matter of fact, we determined this when I was in my mid-20s. People who are in their 50s can often develop something in their eye called ARCUS, which is a buildup of cholesterol, but it normally doesn't happen until you're at least in your 50s. Well, my optometrist found ARCUS in my eye when I was in my 20s and told me I should go see my doctor, and I ignored it. Next year, I went back, and it was a different optometrist, and he told me the same thing. So that time I thought, well, I better pay attention to this. So I went and had my cholesterol tested, and when the doctor's office called me back with my results, they didn't even want me to come back to see my primary care physician. They said immediately make an appointment with a cardiologist. So that was my introduction to genetic disease, but it turns out that I can manage that completely with statins and with fish oil. So I take statins and fish oil, and now my Now my hyperlipidemia is completely under control. So just because you have a genetic disease does not necessarily mean that the answer for that disease has to be genetic in nature, although it is certainly very promising and much energy is going in that direction. One of the things that you've heard a couple of people mention earlier today is that preventing axonal degeneration, and that's why I have this slide back up here, preventing axonal degeneration could potentially be beneficial for both type 1 CMT and type 2 CMT. So again, it's about potentially treating more than one type of CMT with a single therapeutic. So small molecules, why do we call them small molecules? Well, really, it's exactly what it says. They're small. These are molecules that are small. Aren't all molecules pretty small? Well, yes, they are, but of course, it's a matter of relativity. So some molecules are smaller than others. And when you get to a certain size, you call that a small molecule. And above that size, you don't call it a small molecule. Now, you can contrast these small molecules with other much larger kinds of molecules that are sometimes used as medicines that are called biologics. So just comparing and contrasting these here, small molecules are smaller, biologics are larger. Small molecules tend to be produced through traditional chemical processes. Biologics tend to be produced from living organisms, for example, being produced by specially designed yeast or bacteria. You can think about things like antibodies, for example, as being biologics. They are biologics. So when you think about small molecules, you're really thinking about some of the more traditional medicines that you might have in your medicine cabinet. And some examples of that are things like aspirin, things like statins. When we think about biologics, some of these tend to be newer in nature, although not all. Insulin is actually a biologic, and it's been around for many, many decades. But many of the newer medications actually are biologics. And some examples there are Enbrel and Humira. So what you're seeing here is actually the research pipeline from our website. And I know that you can't read this. I'm gonna narrow in a little bit in a moment here, but I want to make a point. And the point here is that there are a little over 20 therapies that are currently being developed for CMT by companies and biotechs. We do not include on this list therapeutics that are still in academic development that have not yet been taken up by a biotech or a pharmaceutical company. So it's incomplete. If you look at what's happening in academia, it would be much, much larger than this. But we really want to follow what's already made it to that biotech and pharmaceutical company stage, because that means it's much closer to becoming a reality for you. So of those 20, more than half are small molecules, which you can see indicated in green here. So even though this is a genetic disease, there is a lot of promise in these small molecules. I'll tell you, you can always find this on our website if you're ever interested in following the progress. We update this about once a quarter. By the way, if we're missing anything on here, I know we had a lot of pharmaceutical companies with us yesterday, and I suspect we do today. So if we're missing something of yours, definitely reach out and let me know. Keith at CMTRF.org. So what are the advantages and disadvantages of small molecules as compared to biologics, gene therapies, et cetera? Well, the advantages of small molecules are that they can easily enter cells. They can get most of where you want them to get in the body to have an effect. They're also much easier to produce because it's just traditional chemical manufacturing. And that can make these drugs much less expensive. They're also more likely to be able to be taken in pill form. You can compare that to biologic medications, and there you're typically looking at some kind of an injection, potentially even an infusion, which takes longer than an injection, and that can be difficult. People generally would like to just have a pill that they can take, and that's much easier, and therefore patients are more likely to use them and more likely to benefit from them. Another advantage is generics. Once these medications go off of patent, they're very easy to manufacture by the generic manufacturing companies as well, and that keeps prices down. What are the disadvantages? Well, they're less targeted than the biologic medications because they can go so many different places in the body. And that means you're going to have more side effects in most cases. And so that can be a concern. Now, you'll notice here that I wrote generics as both a disadvantage and an advantage, and that is not a typo. That's because the competition from generics can be extraordinary. So once as I mentioned, once the medication goes off patent, then companies that produce generic versions can come in and make the generics. That's excellent for consumers who need to take one of these medications, but it creates a real challenge for pharmaceutical companies who have to be able to make a profit if they're going to put the millions, hundreds of millions, frankly, sometimes billions of dollars that it takes to develop a medication. And so the fact that they're so easy to go to generic is something of a disadvantage as well as being an advantage. All right, so just to zoom in a little bit more closely here on some of the specific small molecules, we're not going to give you any detail here. We're just going to take a surface or a survey across the surface here. I would encourage you to visit some of our sponsors and learn more this afternoon, pardon me, from them directly in the Meet the Experts sessions that we have this afternoon, or certainly you can also visit their websites, pardon me, at any time to learn more as well. So in some cases, I think it's important to point out that in some cases, we really don't know how small molecules work. So frankly, sometimes we do know the mechanism and sometimes we don't know the mechanism. A good but surprising example of that is anesthetics. If you're going in for a surgery, that anesthetic will work, it will put you under, but we don't know exactly how or why they work, interestingly. And that can be the case for a lot of small molecules. And I want to be clear here too, none of these medications that you, or potential medications, I should say, that you see on this chart are yet approved for human use because they have not yet been fully through the clinical trial process. And the fact is, we can't know whether any therapy is effective, let alone safe, until it has been through the full clinical trial process that Dr. Motley described earlier. So please keep that in mind. Some of these are very promising, but they are not going to be available yet, nor should they be available yet until they've been proven to be both effective and safe. So in order to be proven, they have to pass all the way through the clinical trial process, including 2 phase 3 clinical trials, as Dr. Motley said. Currently, there's only one medication that's in phase 3 clinical trials, but several that are coming on rapidly behind it. That medication is produced by Pharnext, and right now it has the lead candidate name of PXT3003. It's a combination of 3 small molecules. I also want to draw your attention to the Inflectus medication here. And that's a small molecule that works through the unfolded protein response or the integrated stress response that Dr. Burgess mentioned earlier. That's the medication that actually prolongs keeping the brakes on the process in the cell to keep that cell healthy. In the case of the Inflectus drug, that's a protective process, and so you want to elongate that protection. There's also a small molecule for CMT2A being developed by Mitochondria in Motion. Now, CMT2A is caused by mutations in MFN2, that's a gene that produces a protein called mitofusin. And this is a mitofusin activator that may have some beneficial effects in some forms of CMT2A. There are several companies that are producing or researching Hdac6 inhibitors, which Dr. Burgess also mentioned. Augustine Therapeutics, for example, has one. CKD has one. That's a Korean company, and others are developing Hdac6 inhibitors as well. Now, these Hdac6 inhibitors have a few possible different modes of action, but as Dr. Burgess mentioned, they appear to be active, or at least one way they appear to be active is through allowing axon transport up, so transporting various nutrients and proteins and other molecules up and down that very long axon to keep it healthy. Now, these Hdac6 inhibitors have mostly been studied for CMT2, but I should say that CKD has recently published, and we don't actually have it on our pipeline yet, but they have recently published on a novel Hdac6 inhibitor that appears to be beneficial in a mouse model of CMT1A as well. And moving on, there's a drug screen you can see here being run by Acuristem, one of our sponsors for the convention today. And that's looking at small molecules as well. As a matter of fact, they're looking at more than 3,000 different small molecules for potential treatment in people with CMT2A. So we're eagerly awaiting the final results from that project. I'd be remiss to say that if I didn't say that Acuristem is also studying a small molecule called a PIK5 inhibitor for a more rare type of CMT called CMT4B, which may have benefit by reducing the production of a certain fatty molecule that is overproduced in that form of CMT. Now, this is the plus part of the talk. And I'll be very brief here because you've seen some of this already in Dr. Suko's talk and in Dr. Cleopa's talk, and that is RNA interference. And I put this in as a plus because this is a little bit of a murky gray area whether this is a gene therapy or not. It kind of depends on who you ask. So we lumped it in with small molecules here today, even though it could in some cases be considered gene therapy. Now, we're not going to walk all the way through this slide in the interest of time because we just don't have the time to do it. But what you need to know is that in RNA interference, RNA is delivered into the cell, and then once it is there, it can be used to silence or otherwise modify some of the existing mRNA that's already in the cell, thereby changing which proteins are produced. And there are multiple versions of this. What you're seeing here, we would call risk-mediated. And then what you're seeing on this next slide is extraordinarily similar, but this is RNase H-mediated. So really, it's just a slightly different mechanism in the cell, but largely the same general idea. This is what Ionis first published on, by the way, in 2017. And now, of course, the race is on. So I'm going to end there.